Well, welcome back, everybody. I hope you enjoyed lunch. I hope you enjoyed your time away. For those of you who participated in the breakout sessions, we thank you so much for doing so. Those who did not, you will have an opportunity. Again, so the link was provided in the chat during the first session. And if you'd like to participate in the breakout sessions, you are welcome as well. I want to just welcome everyone back and welcome everyone to our afternoon discussion. So we are going to be taking our focus and bringing it down to the molecular level for the remainder of the day. So we'll have a number of presentations, a panel discussion, and that will lead into our breakout session discussions for round two. So at this time, what I would like to do is turn things over to our session chairs for this afternoon session. And so we have two of our co-organizers, Professors John Yin and Paul Torrens are going to be introducing our speakers for this session and moderating our panel. So welcome back, Dr. Yin and Dr. Torrens.
Welcome back, everyone. I have the honor of introducing our first speaker, Paul Turner, who is the Elihu Professor of Ecology and Evolutionary Biology at Yale University, and he's a faculty member in microbiology at the Yale School of Medicine. He studies the evolutionary genetics of viruses, particularly bacteriophages that specifically infect bacterial pathogens and RNA viruses that are vector transmitted by mosquitoes. Dr. Turner is a fellow of the American Academy of Arts and Sciences, fellow of the American Academy of Microbiology, and he has served as a chair of the Gordon Conference on Microbial Population Biology, as well as chaired the CNRS Jacques Monod Conference on Viral Emergence. In 2019, he was elected to the US National Academy of Sciences. Dr. Turner, we're looking forward to your talk.
All right, well, thanks very much for inviting me. I hope my volume is okay. I will go ahead and share my screen. Okay, do this. Thanks very much for inviting me to participate to NSF as well as to the organizers. So I have more than I could probably say in 20 minutes, so I'm going to get right to it. The first thing I want to emphasize is a familiar slide here based on what we know. The overall goal is to better predict what's going to come next with pandemics and epidemics. So the pathogens that we grapple with now, they are certainly good at infecting more than one host. This is usually the root of the problem. And that other host tends to be us. So this is what causes us great alarm. What I want to focus on today in the short time allowed is that as an evolutionary biologist, moving forward, there are certain topics within evolution that I think are very important here in making future predictions. So as we're reminded from these many examples shown on this slide, this is an enduring problem, but to go forward in the future, what I want to emphasize today is to focus on evolution, adaptation, constraint, and extinction. So here is probably a familiar diagram. I think I borrowed the top half of this from our co-organizer today, John Yin, and elaborated a bit to the lower portion. Even though this afternoon we're dealing with molecular and cellular levels of selection, everything carries through to other levels, I would argue. So a lot of the work that I do in my laboratory concerning emergence and just virus-cell interactions and trying to understand how they evolve is that we think of this as immediately possibly in a more complex scenario where there are multiple host cell types. There's a host that could be a macroorganism host like us that is experiencing the emergence problem, and therefore our own host biology and defenses kick in. Transmission is important. Everybody is focused now on wearing a mask and other ways to reduce transmission in the current pandemic, but there are very many ways that pathogens get around. And then realizing all of this from the lowest levels up to the highest ones, and that there are ecosystem level changes that infect all biological systems, including pathogens. So how do you make sense of all of this? Well, one way is to consider these levels of selection, but to think of themes that cut across them. And here I'm emphasizing the role of evolutionary prediction. So to me, the big thing that we need to do is focus on evolvability, or the capacity to evolve, and how does variation arise in pathogen populations and how is it maintained? Adaptability is different than evolvability, and this is what I should focus on for just a moment for those in the audience who are less familiar. Adaptations are special traits. These are traits that foster the advantage of a variant over those that lack that trait. So that's why they are called adaptations. But as we look at pathogen evolution and try to predict it from an emergence standpoint, we want to focus on the traits that actually foster their success. And a lot of my time spent as an evolutionary biologist studying especially viruses is to not only see what they can do, but what they can't do. And this is called constraint. So as we figure out our world and the threats of emergence, we have to figure out what prevents pathogens from exploiting new hosts such as humans. And the last part of this slide touches on extinction, which is a very real phenomenon on this planet. And yet we have a very, very poor sense of in microbes as well as in pathogens, what they cannot do to persist through time. So the method that I employ in my lab, and then I'm going to focus on for the next few slides, is experimental evolution. This is an approach. A typical design is shown in the lower half of this slide. The main idea is to look at evolution in action and not to recreate the complexities of the real world, because that's impossible inside of the laboratory, but to look at the possibilities of evolution when we control experiments and examine evolution by natural selection and the paths it can take. So this can reveal a lot. It depends on what you want to focus on, but certainly the kinds of experiments that we do focus on molecular and phenotypic variation, the tempo and mode of adaptation. So as I said earlier, adaptive traits are the things that give variants an edge, a fitness advantage, and how fast and how they evolve through time is something we focus on. And the genetic solutions, constraints, these could be plausible solutions or implausible ones. And as we grapple with predicting emergence, we would like to know what are the possible genetic solutions versus the less possible ones. And last, what I said a moment ago is when things don't proceed biologically to persist over time, we call that extinction. And I would say that that's something that's very important in looking at emergence, is the possibilities for it to not occur or to persist. So a very quick case example. I'll go through two interrelated studies that were published by my group just as a way to illustrate how experimental evolution can let us grapple with this problem. So here's a scenario. You can think of environments changing, human impacts especially, just the way that we go about our lives that makes us susceptible to possible emergence events now and in the future. And how this happens is that a pathogen can jump immediately into a new host, or this could be a gradual process. And I would argue that this can lead to very different evolutionary possibilities and outcomes. So if you look around this slide, you can think of human alterations to habitats, humans interacting in, say, a wet market or some very disturbed habitats, interacting with domesticated animals. These are all possibilities for pathogens to jump into humans. And the question is, can this happen immediately such that the pathogen has to very quickly deal with a new host environment, or can it happen more gradually, sort of spillovers that happen while the pathogen is evolving in its current reservoir host, as an example? We wanted to look at these different possibilities, and this was some thesis work done by my former graduate student, Valerie Morley, as well as an undergrad at that time, Sandra Mendiola, who's now a graduate student at Emory. So we thought about this question in the following way. If you consider this graph here, you'll notice that on the y-axis, the proportion of novel hosts in the new environment, under each of these different colored scenarios, it always eventually reaches 1, or complete novel host being the only thing available in the environment. But these different scenarios are the rate at which a pathogen would have to deal with that environmental turnover. Does it happen immediately, such that the existing variation in that pathogen has to deal with that problem immediately, or does it happen more gradually, as in this other extreme shown in red? So we wanted to create an experiment that looked at this. We did so in the laboratory using a virus model system. It happened to be Synbus virus, which is the type virus of the family Togaviridae. It's a positive sense RNA virus, and it's related to rubella virus as well as to West Nile fever virus. Shown in the lower right there is a typical replication cycle for rubella. But what I want to emphasize is the fourth bullet point here is that we used very many test lineages in this experiment because we wanted to look for rare outcomes as well as trends generally across different treatment groups with very many lineages per treatment. And the quick answer is yes, the sudden versus gradual exposure to novel host species does affect virus emergence potential, as shown in this experiment. On the left, if you consider gradual host invasions, which would be more to the right in this graph here. So each of the red symbols is one lineage in our experiment. And during the course of the evolution, we compared these different scenarios of rapid invasion of the new host versus a more gradual invasion into the environment versus what happens if you just let the virus evolve on its current host. But what it's showing you is that this trends upwards, as well as the data become more tightly clustered. That means there's less phenotypic variation at the end and greater adaptation achieved on that novel host when the gradual invasion is allowed. And here on the right, you can find that the lesser genetic variation, as you might expect, follows along with this. So in other words, these populations on the far right are not only achieving higher fitness, but they're doing it in more similar ways. They are not as diverged from one another genetically. So we did a follow-up experiment to this that was just co-authored by Val Morley and me, and this is again from her thesis work. If you consider the Synbus virus genome across the top, one can look through time using deep sequencing in each of her experimental lineages. What are the dynamics of the virus molecular evolution, and is this constrained according to the type of environment? So here on the left is just a snapshot from one of her lineages where the rapid new environment was the thing that was seen by the virus lineage. And what she's seeing is this chatter down here is the viral quasi-species. So this is the variation that's standing in the population. And what emerges from that, if you will, are these different allele sweeps that go through and can go to fixation at the different loci. The black arrow is showing when two sweeps are occurring at the same time. That must be because these mutations are sweeping through at exactly the same moment on the same background. Here on the right, basically I'm showing you for a different population where the introduction of the host is more gradual, the complex dynamics that occur and the greater extent of these kind of cohorts of mutations coming in at the same time. This was shown to be statistically significant. So under gradual host invasion, there's a much greater reliance on mutations acting together through epistasis, it seems, that to solve that problem as single mutations coming in, whereas you don't see that difference here under rapid invasion of a new host. So I'm telling you all this to emphasize that the genetics matter, and you can get some sense of whether the constraint in the system, according to these treatments, is happening one way versus another. And this helps us to build a general prediction. So this paper was published, the last one that I just talked about. I should emphasize that it was supported nicely by NSF. This was published back in 2017. So Val had a graduate research fellowship, and overall, we also received funding from the Beacon Center for Study of Evolution in Action. This paper ended up winning the R.A. Fisher Prize for the most outstanding thesis paper published in the journal Evolution. So the point here is to say that you can get a lot out of these experiments, harnessing experimental evolution in the laboratory to get some general sense of what is happening in a pathogen system if you control it and examine the molecular details. So I went through that very quickly, but I suggest that you can take a look at the paper if you choose to. So to finish up, I'll talk about what other approaches could we use in studying emergence, and there are very many of them. So I'll just have to quickly touch on a few. Here to the left, for example, in my lab group, across the top, we have three so-called model system viruses, a eukaryotic virus, VSV, as well as two phages. And they have different genetic systems as shown in the details there. But more often, we're actually studying the evolution of true pathogens to get a sense of what their evolutionary potential is. We do this safely in the laboratory, but we've done it, for example, with these two types of viruses, chikungunya virus and dengue virus. So here on the right, I'm emphasizing that one can use microbes in the laboratory to study general problems. You can use tissue culture systems like the above right. You can use liquid handling robotics. But what we'd like to build in is more realism in terms of what is happening inside of a macroorganism host, such as a mouse here. It could be interactions of a pathogen coming in and trying to use that new host and competing with the resident microbiome, for example. And there could be other details that could be brought in by harnessing an arthropod vector in these studies. We would like to do high throughput phenotyping in the future. This is something I think would be very important. If you want to look at properties of emerging pathogens, such as viruses, you'd like to get their details very quickly and characterize them rapidly, especially different variants of them. And the existing technology has some limitations to that. So here on the left, it's just a few snapshots from a paper that we published recently on trying to use high throughput phenotyping to figure out what cell receptors a virus is using in encountering cell types. Here on the right, it's a reminder for me to say that emerging pathogens interact with lots of stuff within a macroorganism host like us, our resident microbiomes and viromes, our own host cells, as well as other pathogens that might be present. So we have to keep all of this in mind as we predict how emergence would occur. Measuring phenotypic and molecular rules of virus host breadth is at the root of many things, the emergence problem, as well as what's called virotherapy. How do you harness viruses to do some good? So this is an unpublished data set where if you consider some phages, those are specific to bacteria here on the left, that are pulled straight from nature, what happens if you challenge them to grow on some clinical strains of bacteria that you might want to use them in phage therapy, as shown in the lower right for a young woman who was given emergency phage therapy by our group, led by Ben Chan. He's a research scientist in my group. The dark blue here in this matrix is when the phage is able to successfully infect the challenged bacterium above it. And we know the genetics and the genomics of the system and can kind of see how the cellular level of genetic relatedness does and does not dictate the success of the virus that's able to infect these cell types. If you step away from this data set and can't see the details, you can see it's quite a large data set. And what we're trying to do is take machine learning and computer science approaches to sort through these data sets to accurately predict which of our phages would best be able to cover genotype space in a target bacterium. But that same problem could be turned on its head to think, how can an emerging pathogen such as a virus cover a lot of genotype space for the cells that it is attacking based on their protein repertoire that they are presenting to the world? So this kind of same approach, I think, could harness computer science and machine learning to make better predictions. And the last thing that this might be able to give us some insight into are the constraints, the extreme constraints where lineages will die out through extinction. I would say that we have a very, very poor understanding of this in the microbial world. We are worried about emerging pathogens, but for many, many times, they don't succeed as emerging pathogens because they click into extinction within a new host like us. We would like to know what those background extinction rates are. And this is just a snapshot from a paper that was published some time ago looking at background extinction rates in macroorganisms. We just don't know this for microbes. So the last thing I'll say is that ensuring that we are able to tackle difficult problems in the future means that we want to bring as many people into the workforce to address these problems as possible. So we need to be reminded that diverse approaches come from diverse participants in science. These are some pictures from my lab group through the last 20 years or so, emphasizing that we have lots of people of differing backgrounds wanting to address problems in virus evolution. And that should be what we should keep in mind as we move forward to deal with the emergence issues and predicting emergence in the future. So that's my final slide. I will stop sharing.
Fantastic. Thank you so much for your remarks.
Quite welcome.
We greatly appreciate it. I want to turn it over to the session chairs again to help to introduce our next speaker.
That's me. Trying to find my unmute button here. Sorry, just bear with me.
No worries.
Okay, so it's my great pleasure to welcome Mark Riedel to be our next speaker. Professor Riedel is an associate professor of electrical and computer engineering at the University of Minnesota, where he is also on the graduate faculty of the Biomedical Informatics and Computational Biology group or department. Mark received his PhD and master's degree in electrical engineering from Caltech and his bachelor's degree in electrical engineering with a minor in math from McGill University. He is the recipient of an NSF Career Award, fantastic, and has also held positions at Marconi Canada, CAE Electronics, Toshiba, and Fujitsu Research Labs, and he's going to be talking today about computationally predicting and characterizing the immune response to viral infections. Thank you so very much and welcome.
Thank you very much for the introduction. Thank you for the invitation. I have a short speaking slot here. I'd like to emphasize, first of all, that this work is very much a collaborative work with the Mayo Clinic, with George Vasmatzis at the Mayo Clinic. I think in the audience, you'll quickly infer that this is not my area of specialty, and this is based upon the language that I use. My background is in computer engineering. I jumped onto this topic because of its importance and largely through the collaboration with the people at Mayo, where the experts, I'm the computational person. So I'll be presenting this very much from a computational vantage point. So the topic here is not specifically COVID-19 disease, but I'll frame some of the discussion based upon COVID-19. Our goal is computationally understanding and predicting a specific aspect of the immune system and doing so very aggressively. With regards to a pathogen like COVID-19, we all know that individuals show a very broad range of symptoms, ranging from no symptoms whatsoever, I think that was the case for me, to mild to severe, and of course, a small subset, critical symptoms or even death. And the underlying question that I think makes all of us motivated to study this problem is what causes this variation in disease severity? As we all know, surely, that age, in particular sex and comorbidities, are central. Past exposure to viruses, certainly. But it's the innate differences in the individual's immune system that is our topic of study. And again, this is just one aspect of what influences disease severity, but this is the one that is based upon our genetic makeup, so something that we inherit, and surely this plays an important role. So the goal here is to computationally characterize what's called the cellular immune response. So in a nutshell, this is the first line of defense that we have against viral infections, and also, as I'll discuss briefly, cancer. It's present in all cells in our body, just about, with the exception of red blood cells. And its role is as follows. Given a pathogen infection, a cell, just about any cell, will chop up the foreign proteins into smaller fragments called peptides, and it'll transport these to the cell surface. And once on the cell surface, the peptides, which is the orange-shaped molecule here, can bind to the cell surface receptor molecules called MHC class I molecules. Once bound, the complex becomes a target for the killer T cells, and the killer T cells can come along and kill off the entire infected cell. So this is truly the first line of defense once we're infected by a virus. If this mechanism kicks into gear and the killer T cells kill off the infected cells, then we have a very effective cellular immune response. And this means that likely we show only mild symptoms or no symptoms. But if this mechanism fails, then individual cells then become factories for the virus. They start producing many, many copies of the virus, and likely will have a full-blown infection. So the cellular response then, understanding it, is critical to understanding and predicting the disease severity when a novel virus comes around. I'll just touch upon this later. It's obviously important for vaccine development because vaccines target this mechanism, at least some of them do. And it's important in terms of understanding and predicting the impacts of viral mutations. As viruses mutate, how does this affect the cellular response? And again, cancer immunotherapy. I keep mentioning cancer immunotherapy because that's actually the day job of my collaborator, George. And both of us have kind of jumped into this computational problem largely in response to COVID-19. Autoimmune diseases as well. Now, the core problem, at least from the perspective of a computer scientist, is to predict whether the peptides, shown in this diagram as the blue molecule, will bind to the MHC class I receptor, the cell surface receptor here shown in yellow. And if it binds, then it becomes a target for the immune system. If it doesn't, the immune system can't kick into gear and kill off the infected cells. So the challenge here is that there are many different types of MHC1 molecules. Each individual has up to 6 different MHC1 molecules that present themselves on the surfaces of their cells. Now, of the different parts of our genome, this is perhaps the most diverse part. There are about 21,000 variants of these MHC class I cell surface receptors found in the human population. And no doubt this is because this aspect of the immune response is really important for evolution to act upon. So the scale is many, many different variants of the yellow cell surface receptors. And of the peptides, a virus like SARS-CoV-2 the proteins associated, when chopped up into peptides, translate to perhaps 40,000 different peptides, protein fragments. And so that is the goal, to predict whether those peptide fragments will bind to an individual's MHC1 molecule. So this is, of course, a vast area of study. I only point superficially to some of the work here. Of course, a lot of experimental work spanning decades, where people have tried to understand the structure of peptides and the MHC molecules using experimental techniques. Now, regarding computer science, there's been a fantastic effort to apply machine learning and neural networks to the problem. So an effort called NetMHC. So this is an effort to collect, first of all, training data. So that's the binding strength for all known MHC peptide pairs, and build both neural networks and other machine learning models to try to predict binding strength based upon known pairs. And so here, it's a collection of techniques, including artificial neural networks and support vector machines and other state-of-the-art machine learning techniques. And at its core, it's very useful and widely used, but it is predictions just at the level of amino acid sequences. So peptides are characterized just as a sequence of between 9 and perhaps 12 amino acid sequences. There are 12 possible amino acid sequences given letters of the alphabet. And so the training data consists of labels for the MHC molecules, the corresponding letters, 9, 10, 11, or 12 in length, and a strength. So that's fed into the neural networks. And then the prediction they make is given simply a sequence of letters for a new peptide sequence to try to predict what the binding strength is. So it's a neural network inference based upon peptide letter sequences. And this is terrific, a terrific effort. The neural networks are state-of-the-art, trained as well as can possibly be trained. But the challenge here is that they do make spurious inferences. And this because peptides, for instance, the peptides associated with the proteins of SARS-CoV-2 are brand new. When the virus appeared, scientists had never before seen the particular sequences of peptides. The combinatorial space of peptides is very vast, the number of possible letters we can have in a sequence. And the neural network will attempt to make predictions ultimately based upon similarities in the letter sequences and the strength observed. And that has limitations. So, yes. Yes.
Thank you.
So I will do not much more than introduce the problem in our work. The prior work, well, there's been quite a bit. This is molecular level simulations. It's very sophisticated. But existing software to try to predict things at the molecular level all existing software is general purpose. It's not specific to this problem. And trying out any of this software, we see that it takes hours of days per single peptide pair. But if we have tens of thousands of variants and tens of thousands of peptides, that's perhaps 1 billion combinations, each one of which takes a day of computing time. So for that reason, people judge that it's beyond the scope of the problem to try to simulate this at the molecular level. Based upon an NSF grant, we think we can do better. So we're going to dive right into this fearlessly from the computer science perspective. We're going to do this by developing a highly targeted software. And this highly targeted software will begin not with a general purpose problem of trying to fit one molecule into another, but to start with the peptides correctly designed, placed directly into the cleft, into the binding cleft of the molecule. Then we'll largely explore what are called torsional space moves. So we will just explore the space of atomic bonds twisting. And these are largely the amino acid residues twisting to find the best binding position and hence to predict the binding strength. But the big innovation here is we're going to develop this at scale. We're going to develop targeted software, highly parallelized and deployed on cloud computing infrastructure. And the ambitious claim, which we have not yet demonstrated, is we're going to turn 1 billion days of computing time into just 1 million minutes of cloud computing time. And if we get buy-in, and I think we will, from Amazon Web Services, we can do this in about a month. So that's the scale. Now, there are many challenges which I don't have time to discuss, but one is that we don't actually know the structure of most of these peptides, or in fact, most of the MHC variants. Only about 200 out of 21,000 are really properly characterized, and these tend to be from a specific demographic set, which greatly limits the impact of this work, limits it basically to those of us who are lucky enough to be of Northern European genetic descent. So the challenge here and our solution, we're going to both infer structure and perform the binding calculation in one step. We'll do this by using machine learning techniques, but not on amino acid sequences, but to infer structure starting with similar complexes. Okay, this is my final slide. So the impact here and the motivation, this is a very challenging problem, and we hope to get both the resources to really throw significant computing power at it. But it will be game-changing in terms of what it can predict, because predicting which MHC molecules we have or understanding this can be done easily. It can be done through something that's called HLA typing. It's typically done for paternity testing. So given a knowledge of which MHC molecules an individual has, we can, in principle, predict at least this component of the immune response, and so disease severity for an individual. And then, given different variants of the viruses, we can predict the impact of those mutations on individuals based upon their variants of MHC molecules. And then finally, we can predict the effectiveness of different vaccines, assuming there's a whole range of them that do target specific proteins, and so specific peptides of the virus for different variants for different individuals. And so this can potentially be very impactful. And I'm over time, apologies, I'll stop there and take any questions. Thank you.
Okay, thank you. We definitely appreciate your presentation. Thank you for your remarks, and we look forward to getting even more of your insights during the Q&A, the panel discussion. So apologize for the rush on the slides, but we're grateful to have you as a part of the Q&A as well. Thank you.
I think I have the opportunity to introduce our next speaker, David Van Valen, who is an assistant professor of biology and bioengineering at the California Institute of Technology. His research group's long-term interest is to develop a quantitative understanding of how living systems process, store, and transfer information, and to unravel how this information processing is perturbed in human disease states. So prior to joining Caltech, he studied mathematics and physics, getting dual bachelor's degrees from Massachusetts Institute of Technology, and then his PhD in applied physics from Caltech. He's also had training in medicine at UCLA. So we're looking forward to your talk, Professor Van Valen, I'm sorry.
Thank you. Let me figure out the screen sharing. Are folks able to see my screen?
Yes, we are.
Okay, great. So I'll get started. So thank you for the invitation to speak. Admittedly, so when I received the request, I was a little unsure about what to present. And so today's talk is on some work that my lab has been doing on developing a technology platform for imaging-based reverse genetics. The work is somewhat preliminary, but hopefully you find it relevant and enjoyable. So I'd like to start by discussing some trends in technology. So biological systems are difficult to study because they vary in both space and time, and then also they have large parts lists. And new spatial genomics methods are able to capture both the parts list variation and then also the spatial variation of living systems in the form of images. So some of these methods are captured here on this slide. We see methods capable of measuring gene expression, so spatial transcriptomics, protein concentrations, spatial proteomics, metabolite concentrations, even genome organization. All of these are things that can now be measured with different imaging technologies. At the same time, advances in deep learning have made it easier to extract quantitative information from images. So these methods work by learning how to perform specific tasks from human annotated data. And this is actually a space where my lab has been very active. We've been working quite hard over the last 3 years to develop deep learning methods that can interpret biological images with single cell resolution. And our approach is to really focus on the data side of things and to perform large scale data annotation using the resulting data to power deep learning models. And so here we have 2 examples of, I would consider some success stories from our lab. On the left is an algorithm that can identify and track cell nuclei in live cell imaging movies. And it can even identify when cells divide and construct cell lineages. And then on the right, we have an algorithm that's able to take in images of nuclear and membrane stains of human tissues, and then identify single cells, both the nuclei, but also like the entirety of the cell, and do it with human level accuracy. Okay, so these two trends are making it possible to measure multiple facets of cellular behavior simultaneously, all with imaging. And what I think will likely happen is that as deep learning makes interpreting these cellular images easier, I think it's likely that images will end up becoming a universal data type for biology. And I think there are advantages to this world beyond just having diverse integrated single cell data sets. The one that's actually the most appealing to my lab is just having a lower marginal cost of data for single cell measurements. The economics work out that imaging, as far as the marginal cost, is much cheaper than sequencing. Okay, so as I said earlier, for the last 3 years, my lab has sort of been contributing to these technology trends, but we're also very interested in using these trends to explore how viruses and their hosts talk to each other. And we are particularly interested in how information from and about the host influences the outcome of viral infections. One model system that we've been studying is a latency decision in bacteriophage lambda. So shortly after a bacterial cell, E. coli to be specific, is infected by this virus, a decision is made to either make more viruses or to become latent, and so in that case, the viral genome ends up integrating into the host genome, and ends up sort of following along for the ride every time the host cell then replicates. And so this decision-making pathway on the virus side has been fairly well mapped out, but just due to technical reasons, there haven't been systematic studies of what happens on the host side. And so the extent to which this is a virus-dominated decision, a host-dominated decision, or a mutual decision, I'd say it's not really that clear. And so we've been studying the role of the host in this decision systematically using imaging and reverse genetics. And so the experiment that we've devised and executed sort of goes as follows. We've created a reporter virus that reports on the infection outcome. Infections that result in more viruses being made cause cells to turn green because GFP is being expressed. And infections that result in latency cause cells to glow red because mCherry is being expressed. And so using this reporter virus, you can then infect a library of non-essential host gene knockouts. And so you can imagine that if a host gene was important for this decision, if you remove it, you would cause the decision-making outcome to shift one way or the other. Assume that your initial background condition was roughly 50-50 either way. So again, if a gene was important, you'd expect either most infections to end up being active, producing more viruses, and hence most cells would be green, or it would shift things all the way towards latency, and hence most cells would be red. And so we've actually performed such an experiment. So so here's a movie showing like what such a reporter virus does. So the green cells are the ones that end up making more viruses. The red cells are the ones that end up being latent. And so we've actually performed this experiment. So we performed high throughput imaging of infections in over 3,000 different knockout strains. We used deep learning algorithms to analyze the resulting images with single cell resolution. And so then we can quantify the outcome of each of these of infections in each of these different background strains. And so the result of this experiment is a collection of over 100 different host genes whose removal results in the infection results shifting strongly towards either being active or strongly towards being latent. So most of these genes actually weren't known to play a role in this decision. Some of them were, and they naturally appear in the result in our data. And so the point of this slide isn't really to focus on any one gene, although there are interesting genes and interesting gene families. So there are ties to host metabolism, a collection of host transcription factors pop up, and we have some preliminary data showing that these host transcription factors are actually functional and relay environmental information to the decision. But rather the point is sort of twofold. One, to show that these approaches can generate a lot of data, a lot of rich data. And the second is to say that it's actually possible to map out which host virus interactions actually matter to a relatively complex phenotype entirely with imaging. Okay. So one dream that we have is to actually do a similar experiment for mammalian viruses. And there's a lot of reasons why one might want to do one might want to do this. And I say a big driving factor for us is just like this entire field of quantitative virology has produced gorgeous data on the life cycle of mammalian viruses. So here, this is a data set from a paper that was published last year in Cell, showing an RNA virus replicating shortly after it infects a cell. And so creating for experiments like what I just described for mammalian viruses, creating reporter viruses, doing the genetic engineering, collecting movies, even collecting movies at high throughput, those aren't really what we see as the bottlenecks. The real bottlenecks are actually applying perturbations to the host. And this is really just an issue of scale. Mammalian organisms have roughly 10 times more genes and hence roughly 10 times more parts than bacteria. And so if you want to do these sort of systematic studies, it just makes it more challenging. Technically speaking, advances in CRISPR make performing these genome-wide perturbation studies possible, but interpreting them with imaging has its own unique challenges. Arrayed screens are more expensive. So by arrayed, I mean where basically you have a collection of 96 well plates, and each well ends up having one perturbation. These arrayed screens are expensive, and they also frequently require a fair bit of laboratory automation. Pooled screens are easier, but there is a technical challenge, which is you need to be able to identify which cell, when you're imaging, you need to be able to identify which cell received which guide RNA, and you don't know a priori because all of your perturbations sort of exist, in this pool collection. Okay. So methods for imaging pooled CRISPR libraries exist, and they work by tagging each guide RNA with a sequence that's called an optical barcode. So an optical barcode is a DNA sequence that can be decoded with imaging. And this decoding step is sort of what makes this feasible, but the existing approaches are sort of difficult to implement, and then also not that scalable. They're all effectively some version of sequencing on a microscope. One method that's produced by a lab at, well, there are two labs, one at the Broad, one at Harvard. One method uses optical barcodes that rely on multiplexed RNA fish to be read out. Another approach uses a rolling cycle amplification followed by in situ sequencing to be read out. And these experiments are both, to actually use in practice, the experiments are both technically challenging and also require doing a lot of imaging to read them out. And so we've been working on an alternative approach which relies on creating distinct spatial patterns within cells that have multiple colors, and then using those as the optical barcodes. And the combinatorics of this alternative approach are more favorable. And just doing some back-of-the-envelope calculations, we think it's possible to be able to barcode a genome-wide library and then have the decoding step be done with a single round of 3 to 4 color imaging. Okay,
just
so
a minute left, Dr. Vandella.
yes, I'm almost done. It's the last slide. So the approach we've taken to actually developing these barcodes is through CRISPR. In addition to providing a perturbation, we've been using CRISPR to paint different repetitive regions in the genome with multicolor RNA tags. And so to date, we found approximately 25 different regions within the nucleus that, when labeled in this fashion with CRISPR, give rise to distinct spatial patterns. And our preliminary data shows that we can distinguish some of these patterns specifically using deep learning. And so I think this is some very exciting proof of principle data showing that this approach is both viable and possible. And so while there's a lot of work to do, performing more benchmarking experiments, constructing and actually testing such a library, I think this technology, like when complete, will really enable systematic studies of host virus interactions entirely with imaging. And I think it's also an example of how one can use both algorithms and technology to develop better, more scalable experiments. Okay, so I think I'm out of time. And so I'd like to briefly thank the members of my lab who actually did this work. So the virus work was done by an undergraduate student who worked with me when I was a postdoctoral fellow at Stanford, Nicholas Quach, and then also a technician in my lab, Geneva Miller. The barcoding work was done by a fantastic graduate student, Morgan Schwartz, an undergraduate student, Jonathan Soro, and also our lab manager, Edward Powell. And lastly, I'd like to thank our funders, most notably the Kersey Foundation, the Rita Allen Foundation, and the Susan E. Riley Foundation for supporting this work. And with that, I will stop talking.
Fantastic. Thank you so much. Okay, we're going to move to our panel discussion at this point. So we're going to invite all of our speakers to rejoin us, virtually rejoin us in order to spur some discussion. There has been some Q&A already in the chat, and we're grateful for that. I'm gonna turn it back over to the session chairs to moderate our Q&A. Thank you.
Maybe I can go ahead and start off. I did put a question in the chat. I think Professor Turner responded to it, but I'm not sure all our participants were able to see his response. So I'll just repeat it. The virus variants of SARS-CoV-2 are creating broad concern over more efficient transmission and or severity of disease by this virus. So how might the experimental evolution methods that you've developed be harnessed to give insights into these challenges specific to the current pandemic?
Yeah, that's a great question. So the role of experimental evolution is to look at possibilities. So these variants could be put side by side with the so-called classic variant, the one that actually launched the pandemic, to look at very many questions, to compare their evolution and adaptability, etc. So of course, the main problem is that it's dangerous to do that. It's not that easy to do that under an easy biosafety level. So this gets lumped in with experiments that are very difficult to do because you have to have the right infrastructure. It's not like any laboratory could do this. So there are other approaches that could help. You could take the what looks to be one of these variants on the exterior, but the genome would be something that is of a kind of a chassis of a more trusted, safer virus. But that doesn't get you very far because it's really that genotype that would change, and we're not so interested necessarily in only the proteins in an unchanging state. So I guess my final answer to this would be, this is how we can harness close relatives of pathogens and look at them more safely under laboratory conditions that are not so extreme. I think that is just what we need to do. You know, you can examine experimental evolution of true pathogens. We do it in my laboratory. It requires the right infrastructure. I would be remiss if I didn't say that that's the most powerful way to do it, but we have to be careful about that. So it's a combined approach, what's practical versus something that could be a little more extreme and not so easy to control.
Great, thanks.
John, would you like me to ask another question?
Go ahead, sure, why not?
All right, so this one is for Professor Riedel. Actually, he answered it, but the answer only went to the panelists. We have two different systems going on here, so give an opportunity to answer the question he's already answered. So the question is there any, oh no, that's the wrong one. Are you developing your own simple torsion-only molecular mechanics force field or using existing force fields?
Yeah, that's a great question.
Yeah.
So we're using existing force field calculations. In essence, the binding aspect is not the innovation here. The innovation compared to approaches like molecular dynamics is, instead of spending a lot of time to find the optimal binding configuration, instead of spending a lot of time moving the peptide in three-dimensional space and having the software basically through random moves try to find where the peptide should fit, we're going to start with the peptide in the correct location, in the groove where it can fit and properly aligned. And then only explore through torsional moves, basically the residues, the amino acids, how they twist around on their bonds to try to see what is the optimal set of torsional moves that will give you the best binding energy. So it's essentially an optimization in the strategy, but we think it can buy us a huge amount of computing time. If that's the question. But we're not taking shortcuts in the sense that we're not approximating the binding strength by only looking at one aspect of the binding action. We're going to use all the same sorts of binding calculations that the existing software uses.
Great, I can follow up with a question that goes both to Mark and David. From our audience. Is there a scarcity of labeled data? So how would you describe the main computational challenge in your problem? Is it scale, noise, scarcity, dimensionality? Do you use any simulations in conjunction with data as well?
So I can speak to imaging data. So I think for imaging data, there is a very severe lack of annotated training data for these purposes. And I'd say that's actually like largely, that's one of the problems that my lab has been focusing on solving. It turns out that so there are different ways of annotating imaging data, but it turns out that what's the most useful for biologists is actually the most difficult type of annotations to create. So most problems are that biologists want to be able to do. They need dense pixel level labeling. So for the examples I showed in my talk, what you need a person to be able to do is to go through all these images and then, for every pixel, say like, okay, these set of pixels belong to cell number 1, cell number 2, cell number 3, and whatnot. If you're dealing with movies, then you have to do that for every frame. You need to have some way of linking things together over time. And, for some of the data sets now, they're becoming higher dimensional. So either 3 dimensions in space, 2 dimensions in space, 1 in time, 3 dimensions in space, 1 in time, 1 in time. And then for the multiplex imaging methods, there you can have, literally dozens of different colors, all of which are meaningful. And figuring out how to label data that is of that type of complexity is quite challenging. I'd say like we've spent a lot of time and also a lot of money trying to figure out how to do that. And I think like we're getting close to having some solutions. So I can tell you like about a couple success stories. So for tissue imaging, we embarked on a large-scale annotation project so that we could have models that can do single cell segmentation, both nuclear and whole cell in tissue imaging data. And so as part of that, we ended up labeling, I want to say, around 1.3 million cells across a wide variety of different tissue images. Figuring out how to do that in a way that is both scalable and affordable was non-trivial. I'd say like these are projects that, this sort of project is something that's relatively unfunded. I think I'd say it's the elephant in the room when it comes to machine learning methods. People are sort of dazzled by the fancy math and the cool programming, but these sort of tasks where it's like, hey, we got to figure out how to label, like 1 million, 2 million, 3 million cells. That's less exciting, but that's actually what you need to get these things to work. So we've figured out like a human in the loop approach to doing scalable annotation. It makes things, more affordable. I'd say if we had to do such a project again, generating 1 million labels would cost around $20,000. And the models that come out the other end, when they're backed by large amounts of diverse data, they're just they're more accurate and they're more useful. On the in-distribution data, so tissues that look like the tissues that represent our data sets, we've been able to demonstrate that we can achieve human level accuracy.
That's great.
Yeah.
I do want to give.
Sorry, I'll stop talking. But it's a big challenge.
No, that's great. I also wanted to comment before we moved on that it is precisely the elephants in the room that we are here to talk about. So I appreciate that comment. These are the sorts of things people don't want to talk about. They're not always the sexiest part, but they are important. So we definitely want to highlight those, and I really appreciate that. We should hear from Professor Riedel.
Yeah, so I can keep my answer short. It's definitely scarcity of data that's the biggest problem, and the scale of the computation. Scarcity of data because the experimental data only includes a very small set of the molecular structures. And so what we're proposing is that we're going to simulate it on a vast set, and yet we don't have the actual 3D structures for most variants. But it's also the scale of the problem, because I think inferring the structure might not be that difficult, but to do it at scale and to do it accuracy is a problem. And the scale of the computation is such that just using existing software doesn't work. And an elephant in the room here, it's not a particularly glamorous work to rewrite existing software, some of which was written decades ago, and run it on GPUs or high performance clusters. That's exactly our expertise. It might not lead to immediate high impact publications because you've just re-implemented software and techniques that have been around for a while, but I think we can do it better. And so that's the challenge, to convince funding agencies that we can do it better before we've actually done it, even though we're not really inventing new science here.
Great. Paul, you want to go ahead?
I'd love to. I think we're at a break.
Oh, yes. I think we're out of time. That was a fast Q&A,
Yeah.
but very informative.
No, I'd love to keep on talking, but
The great news is we're going to continue this conversation in the breakout rooms.
there you go. Thank you, Manuel.
So we know that there are still outstanding questions, and that is the nature of this research and this time that we have here in human history. So we look forward to inviting all of you to join us in the breakout sessions to continue this important conversation. I want to again just thank all of our panel members for briefing us on the exciting work that you're doing in your field, sharing your insights, and also helping us to ask new and different questions as well. So thank you all again. And now for those who are going to be joining us in the breakout sessions, again, I'm going to ask you to make sure that you go ahead and drop off of this webinar so you're able to join the Zoom meeting. Make sure that you have the link. You should have either received it in an email or that link was provided. I believe we can provide it here in the chat once again. For those who will not be joining the breakout sessions, we will resume back here in the plenary session for a closing discussion once the breakout sessions have ended, and that is scheduled for 5:05 Eastern time. And so for those of you who are not participating in the breakouts, if you'd like to take some time away, you can feel free to meet us back here at a little after 5 o'clock. If you're in the breakouts, please try to join as quickly as you can so we can get you into your respective groups, and we will see you there. Take care, everybody, and thanks again.